Vitapcr is a new rapid molecular in vitro diagnostic system for point-of-care use. It uses real-time PCR technology for the qualitative detection of SARS-CoV-2 viral RNA in 20 minutes. Vitapcr is a highly effective and efficient solution for labs, hospitals, emergency rooms, triage tents, mobile structures, nursing homes, and entry points like airports, train stations, etc. Vitapcr is easy to use, rapid, versatile. To start the test, you will need a Vitapcr SARS-CoV-2 assay kit, a rack, a pipette for either fixed or adjustable volume sampling able to dispense 30 microliters, a filter tip for the pipette, a nasopharyngeal or oropharyngeal swab for collecting the specimen. Attention! It is recommended to use flocked or synthetic fiber swabs with plastic shafts. It is important not to use swabs with wooden shafts or calcium alginate swabs as they might contain reagent inhibitors. Do not use swabs that have been inserted in test tubes containing UTM or VTM transport medium. Turn on the instrument by pressing the button in the bottom right-hand corner. If you wish to save the test results on a USB key, insert it into the rear port of the instrument before starting the test. Use only USB 3.0 FAT32 type keys. Using the touchscreen display, tap on Select User ID and select Admin. Tap on Enter User Passcode and enter the code. Tap on Login to confirm. Tap on Run Test to start the test. Using the barcode reader located at the bottom of the instrument, scan the barcode on the aluminium pouch containing the reagent tube. Tap on the Next button and confirm with OK. Using the barcode reader at the bottom of the instrument, scan the patient's ID or type it on the display. Tap on the Next button and confirm with Confirm. On the next screen, tap on Skip Instruction to start preparing the sample for the test. Alternatively, you can follow the step-by-step instructions of the entire procedure on the display by tapping on Next. Write the patient's ID and the sample's date onto the label of the lysis buffer vial. Place the lysis buffer vial into the rack. Withdraw a sample from the patient using a nasopharyngeal or oropharyngeal swab. It is recommended to conduct the test immediately after taking the swab. If this is not possible, samples can be stored in the lysis buffer at room temperature for 1 hour, at 2 degrees Celsius to 8 degrees Celsius for 10 hours. If instead the sample is not collected on site, the swab can be transferred into a clean, dry, well-sealed test tube and stored at -20 degrees Celsius for 24 hours. Uncap the lysis buffer vial and keep the cap. Insert the swab into the lysis buffer vial and rub the swab against the wall of the vial 15 times. Then discard the swab. Recap the lysis buffer vial and shake it 10 times. Place the lysis buffer vial back into the rack. Open the aluminium foil pouch containing the reagent tube. Make sure that the freeze-dried reagent is sitting at the bottom of the tube. If not sitting at the bottom of the tube, tap the tube to make the reagent drop down. Then place the reagent tube into the rack. Open the plastic bag containing the reagent tube's cap. Place the reagent tube's cap onto the rack, taking care not to touch the top of the cap. Open the reagent tube by removing the upper seal. Take the cap off the lysis buffer vial. Set the sampling pipette to 30 microliters. Install the filter tip in the sampling pipette. Press the plunger and insert the sampling pipette into the lysis buffer vial. Slowly release the plunger and withdraw a 30 microliter sample avoiding the formation of air bubbles. Insert the sampling pipette into the reagent tube. Press the plunger to transfer the sample withdrawn into the reagent tube. Slowly pipette several times, avoiding the formation of air bubbles. Withdraw the pipette from the reagent tube and discard the filter tip. Close the reagent tube with its cap, pressing down until you hear a click. Make sure that the cap is firmly pressed onto the reagent tube. If air bubbles form inside the reagent tube, shake it 3 times. Press the blue button at the top of the instrument to open the door. Insert the reagent tube into the hole and close the door again. Using the touchscreen display, tap on Run to start the test. Wait 20 minutes for the procedure to finish. At the end of the test, the result will be shown on the display. The target RNA sequence to be detected is found in the region of the virus nucleocapsid N gene specific to SARS-CoV-2. As additional reference information, the test also provides the results of the detection of the conserved region, which is also found in the N gene of SARS-like viruses, including SARS-CoV-2, SARS-CoV, and SARS-like bat coronavirus. The test also provides for an internal control capable of detecting the presence of HBB, human beta globulin, which makes it possible to check whether sampling has been carried out correctly and monitors the possible presence of factors that could inhibit the PCR reaction. The automatic interpretation of the results can give the following results: positive with identification of both SARS-CoV-2 and SARS-like RNA, positive with SARS-CoV-2 identification only, presumed positive with identification of SARS-like RNA only, negative, invalid test. By tapping on the button Fluor Curve, it is possible to display the graph of the amplification curves. All results of the tests conducted are saved in the instrument's memory. It is possible to access the archive of saved tests on the home page. At the end of the test, press the blue button at the top of the instrument, open the door and take out the reagent tube, which must be disposed of.